Good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Viral Vector Manufacturing. Are antiviral defenses the missing link to scalability? My name is Aisha Rashid and I will be your X-Talks host for today. Today's webinar will run for approximately 45 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this time, I'd like to thank Verrica Biotech for putting together the content for this presentation. Verrica is the first company to develop and commercialize viral sensitizers, or VSEs, as a solution to the challenge of antiviral defenses in the manufacturing of viral vectors. VSEs are patented small molecules, including known drugs and novel synthetic compounds that transiently attenuate antiviral pathways in cells, and thus can increase both virus production and therapeutic efficacy of viral medicines. Verrica's VSE platform evolved from research conducted by Jean-Simon Diallo, Verrica's scientific founder and CEO, and a world-recognized leader in oncolytic viral therapies at the Ottawa Hospital Research Institute. Dr. Diallo and his team have developed unique high-throughput virology methods that have to date identified over 100 VSEs. And now, without further ado, I'd like to hand over the mic to our speakers, beginning with the moderator of the session, Benjamin McLeod, who is the technical lead at Verrica Biotech. Ben, you may begin when ready.
Thank you, Aisha. Thank you, everyone, for coming today. My goal here is to be the moderator of this webinar. So the way that this is going to be working, we're going to, first of all, introduce the speakers. Then we're going to be essentially having a quick presentation on what antiviral defenses are, what they look like. Then we're going to move into the bulk of the webinar, which is a fireside chat, diving into some of the unique details related to antiviral defenses and how they can impact manufacturing. And then we'll finish it off with a Q&A. So again, if you guys have any questions with regards to what we're talking about today, by all means, feel free to put those in the chat. So without further ado, I'll just introduce the two speakers, starting with Dr. Jean-Simon Diallo. So he's the CEO and founder of Verrica Biotech. We're very pleased to have him. He's been studying antiviral defenses for well over a decade, so he's a bona fide expert in this. Secondly, we have our VP of Scientific Operations, Dr. J.D. DeYoung. He has a PhD in virology. He has been heading up Verrica's R&D efforts for several years now. Gentlemen, looking forward to a really great discussion.
Thanks, Ben.
Thanks, Ben.
Excellent. So just before we start, antiviral defenses are a topic that's gotten a little bit more traction and attention over the last couple of years, but we're just going to have a quick poll question just to gauge your understanding of antiviral defenses before we start. So we'll just quickly bring up the poll question. So the question is, how aware are you of antiviral defenses in your production system? Very aware, moderately aware, or not aware? And for any of you who are not aware, we would appreciate it. We appreciate that you're here. So yeah, please, by all means, we would appreciate if everyone can quickly answer this poll question. All right, we'll give it another 10 seconds or so. And then we'll be going through the results together. So by all means, if everyone can participate, it'd be greatly appreciated. Excellent. All right. So we've got a nice diversity where most people would be moderately aware of them. That's great. For the 29% of you who are willing and humble enough to say that they didn't know that much about antiviral defenses, that's great. Thank you so much for coming. And we're hopeful that we'll be able to clear that up for you. Excellent. All right, so we'll move on to the meat of the webinar, which is going to be, first of all, that introduction on antiviral defenses, and then a fireside chat between JS and JD. So we're going to start with JD, who's going to be providing a bit of an overview on what antiviral defenses are and why they are important.
Yeah, thanks, Ben. So this is just to level set everyone and kind of contextualize the conversation going forward. And this is the concept of the innate antiviral defenses within the cells. So generally speaking, when we talk about the immune system, we talk about the two different arms of the immune system. You know, the one that most people are familiar with is the adaptive arm. This is the ability of our body to basically educate itself against a very specific pathogen. We get antibodies and T cells and all that good stuff that underpins our modern vaccination strategies. But the other arm of the immune system is the innate immune system. And one of the components of that innate immune system is the fact that every cell in my body and everyone else's body who's participating in the webinar today actually has the intrinsic ability all by itself, irrespective of that adaptive immune response, to fight off viral infection. And this is part and parcel of what keeps us healthy and alive today. Those same pathways that are in our cells within our body remain partially, if not fully intact, in the cells that we're asking to create or generate these medicines, these viral-based medicines in bioreactors. So you take the example of HEK cells. They most definitely have some intact innate antiviral pathways, and JS is going to dive into that a little bit more detail in a second. But even vero cells, for example, traditionally thought to be immune incompetent, and that is certainly true. They do not produce type 1 interferons. However, they do produce type 3 interferons, the lambda interferons, and definitely mount an antiviral response. So now that we've talked about that a little bit and how it applies in the bioreactor situation, I guess one of the questions would be, well, how do cells respond? How do they know when to mount these types of responses? And it really comes down to specific molecular signatures that we call PAMPs, pathogen-associated molecular patterns. These are unique molecules to invading pathogens, so kind of non-self molecules that our bodies and our cells have basically evolved over time to detect. So there's a whole suite of detection molecules, ready and kind of willing and able as a frontline defense to detect incoming pathogens. This then triggers a whole host of signaling pathways within the cell that basically mounts this antiviral state through a bunch of effector molecules that get developed. And so these PAMPs kind of come in two different flavors as we talk about it. One would be incoming virus or even incoming transfection reagent, because a lot of these sensors are actually sensing foreign nucleic acid. So if you're RNA virus, DNA virus, or even if you've got a complex of plasmid DNA that's coming into the cell, that is sufficient to set off those bells and whistles and really start mounting that response. So that's wave number one. But there's actually a second wave of PAMPs as well. And this is the byproducts of the assembly and replication of these viruses. So maybe some double-stranded RNA is being made, some unique molecular signatures, all the proteins that are making up the capsid. Again, this is a second wave of PAMPs that sets off kind of those bells and whistles within the cell. So really what I'm hoping you can appreciate out of all of what I said there is that these innate cellular antiviral defenses are definitely present within bioreactors and within the cell populations in those bioreactors. They're quite complex and intricate. They've evolved over, thousands and millions of years to detect these. And they're likely playing a key role in blunting both the yield as well as possibly the quality of the vectors that come out of these bioreactors. So hopefully that level sets everyone and kind of gives everyone a flavor of the innate antiviral response. Oh, I think you're muted.
The classic. All right, thank you so much, JD. We'll move on to JS.
Yeah, so thanks, JD, for that introduction. I'm just going to follow up on that. I think you touch on something that's really important, which is those different waves of signaling that can happen in the cells. there really are those three components: the sensors, the signal transducers, and then the effectors, which really have the antiviral effects at the end of the day. And again, you know, for years, I think people have more or less ignored this in the manufacturing context because of you know the understanding that we had at the time. JD touched on the vero cells not having type 1 interferon. Well, at the time when that was discovered, you know, that's all we knew about the antiviral systems. We didn't know about lambda interferons and type 3 interferons. So the knowledge of the antiviral defenses has really skyrocketed since those early days where these manufacturing cell lines were selected, and generally have been selected for their ability to propagate viruses more efficiently than your typical normal cell. And so, you know, only now are people starting to really understand and look at the antiviral defenses in their manufacturing processes. And, you know, recently a paper was published by Eric Barton's group at Pfizer, where they actually looked at the transcriptional response to AAV production in a 50-liter bioreactor scale following triple plasmid transfection. And so what you can see here in the graph on the right is really something that basically the figure that they published, which really shows that initial response to transfection. So that's really a stress response. And then that's followed by a secondary response that really is associated to the regulation of a variety of different inflammatory genes and antiviral response genes, as well as other nucleosome and metabolic process genes. And really that phase of it is actually associated to the growth and the packaging of the AAV vector. And so really at the end of the day, what you're looking at is a temporal and kinetic response that occurs in this particular manufacturing system. And while this is the case for AAV, surely this also happens for other viruses that you're producing, such as lentiviruses or replicating influenza viruses. The only difference is going to be how did you produce the virus, which cell line did you use to produce the virus, and how quickly is your virus growing in those cells, right? And how is that end signal of the antiviral defense being integrated across space and time in your process? So it's obviously a very, very complex thing. So, you know, the intricacies of these systems are a challenge, but they're also an opportunity to intervene to actually improve the yield of the viruses, because of course, you know, the genes that are really going to be important here are going to be those antiviral effector genes, because they're actually going to physically destroy the viruses, right? They're going to, you know, we're talking about genes like, you know, ApoBec, RNase L, you know, the ubiquitin proteasome system, ISGs like ISG15. These are things that are actually going to lead to the destruction of the virus, that will actually mutate, lead to mutations in the viral genome. So AAV is a, you know, is a hot topic these days, because of course there's so many gene therapies that are based on this vector, and there's a lot of conversation around the packaging of AAV vectors and the fact that during production you often have these empty particles that are devoid of genomes, which will lead to an immune response if you don't get them eliminated out of your final product. And obviously these are things that you don't necessarily want as part of your gene therapy. But even when the particles are full, they could be full of junk. So, you know, part of that is how we make AAV, for example, using different plasmids. You can have incorporation of plasmid sequences in your AAV, which is obviously not good. And you know, I'm reminded of a presentation at the ECI conference for cell culture engineering in Cancun about a month ago, where Dr. Guanping Gao, you know, a pioneer in this space, really presented, you know, very interesting information around the packaging of these, you know, aberrant particles of AAV. but also how you could use things like proteasome inhibitors to improve the productivity. Again, thinking about how we start targeting these antiviral defenses. And so obviously the quality of your viral vector is possibly being impacted by these antiviral defenses. Even when you have fully packaged genomes, they may be mutated, right? Some of these proteins, like Appleback proteins, for example, include cytosine, there's cytosine deaminases, so they actually can mutate the virus, and you might be able to see full particles by PCR, but actually they're not transducible, or they won't transduce your gene of interest properly. And so these are all factors that can come into play, and ultimately why we need to really focus on some key quality attributes to the viral vectors that we're producing at the same time. So, again, we're really starting to see the evidence of antiviral defenses in manufacturing. The work by Dr. Barton's group is the first to be published on this in AAV. But again, at that ECI conference, I also saw some very nice work from Sandra Klausing at Sartorius, where she essentially showed the same thing as the Pfizer team, but also showed that not only did this occur in these HEC293 cells, but also change from subclone to subclone of HEC293, which is another thing that that most people don't think about. The variety of subclones of these cell lines, we call them all the same name, but they're very different cells, and that's an important consideration when you're thinking about antiviral defenses and how they work.
Very good. Excellent. So you've spoken a little bit about this, so about the quality metrics already, but maybe we can dive a little bit deeper into this and just spend a little bit less time on it. So this Pfizer paper, which is really quite an interesting paper, has postulated that antiviral defenses could be modulators of recombinant AAV yield and quality. Let's again just kind of walk through those different quality metrics that are standard in the industry, and then also would antiviral defenses impact quality metrics that aren't even being tracked. We've talked about this a little bit already, so we can probably move through this section a little bit faster.
Yeah, and I think JS really set the table very nicely with the description of some of the effects that this antiviral response could have on your end product. And we're starting to see in the industry as a whole, this won't be a surprise to a lot of the folks in the AAV space in particular, that the conversation is really evolving beyond just VG per mil titer, right? That was a good starting point to really measure the productivity of any given process. But now you're starting to see not just the full empty, but the full empty partial conversation, really coming to the table. Obviously, there's been some really nice advances in the different techniques available to measure this, making things a little more accessible than AUC. You have some mass spec based techniques, or HPLCs, and things like that. But again, the question is, what are we missing? Because at the end of the day, and the JS touched on this very nicely, is something might appear full, but what is it full of? Are you getting some of that cross packaging of the plasmids? Or even more difficult to detect, are there small indels or point mutations within the genome, so it looks completely like it's supposed to, but the transgene is not the therapeutic that you want it to be? And so this is where things such as NGS, I think, will start coming into play, where you're going to really get a good snapshot of the heterogeneity within that population. And then beyond that, beyond the genome, what parameters are really affecting the capsid? I think this is a really interesting question. JS again touched on the ISG15, ISG-elation of the capsid, or any of these post-translational modifications of the capsid. How is that influencing the potency of the vector? So I think it'll be really interesting to start having conversations around those critical quality attributes, and then more importantly, getting the analytical techniques. So there's a biology component here, really understanding what contributes to the potency of the vector, but then also having the proper analytical techniques to have a look at it.
Very good. So JD, one of these quality attributes that's of a lot of interest to me are these partially full capsids, something that the industry hasn't really looked at too much until recently. Could you speculate about how antiviral defenses could be producing partially full capsids?
Yeah, so this again goes back to those effector molecules that we talk about, right? So this cascade of sensing and then really getting the cell in that antiviral state. And a lot of these effector molecules, as JS alluded to, they're really there to attack and modulate the key building blocks of the virion itself, right? So you get these virions self-assembling, in certain compartments within the cell. But at the end of the day, they're protein, nucleic acid, and possibly lipid if you have these envelope viruses. These effector molecules are really honing in on those key pieces and either truncating them, destroying them, mutating them, tagging them certain ways, tagging them for destruction. I mean, there's even the tethering, right? That's going to grab onto the lentivirus and not allow it to escape the cell. Like, you can't make this stuff up. It's really cool stuff. But there's this whole suite of molecules there that are there to attack the building blocks of the virion. And this is what we think could be contributing to, and I don't want to make this all AAV specific, because this certainly applies to replicating viruses as well, really contributing to defective genomes. I think that's probably the conversation we should be having, rather just saying partial, full, but really just defective genomes or defective particles coming out of the cells.
Excellent. Very good. All right. Well, if that, in that case, just before we move a little bit further on into the fireside discussion, we're just going to have another poll question. So if we'll pull that up for just a second here. What is the most significant challenge, in your view, in viral vector manufacturing? Is it functional viral titer yield? Is it the time efficiency for a target yield? Or is it cost-effective production? And when we were doing a run through yesterday, JD said, "Oh man, I wish I could pick all three," but unfortunately, we can only pick one today. So if you could pick one, that would be great. And if everybody could participate, that would also be awesome. All right, we'll leave that up for around another 10 seconds. And then we'll look at the results again together. All right, so we've got a narrow winner, functional viral titer yield, and I think that really is one of the best answers. If you're able to make it faster, but the quality is lower, then that's not going to be too useful. Obviously, costs are going to be a major component as well, but even if you can drive down costs, but the functional viral titer isn't all that great, then it's going to challenge the overall efficacy of your therapeutic. So interesting to get those perspectives. Thank you so much. Excellent. All right, so we'll move on to the second half of the fireside chat here. The one thing that I wanted to talk about next is, why as an industry should we be concerned about antiviral defenses? We've looked at recombinant AAV. JD has briefly mentioned replicating viruses, but why should the industry as a whole be concerned about this?
Well, I think the survey kind of says it in a way, right? Yield and cost are both linked together, and I think if there's any chance that antiviral defenses are decreasing our yield, whether you're producing a gene therapy vector or a vaccine, I think we need to be really aware of it and also proactively dealing with it. Because really, at the end of the day, the complexity of the antiviral defenses is probably the biggest challenge that we have. Because at the end of the day, the antiviral signature that is experienced in the cell that you're manufacturing in is really the product of the specific genetic makeup of the cell, genetic and epigenetic, I should add, as well as the virus that you're trying to produce very specifically. You know, some wild type viruses have genes already included that downregulate certain antiviral defense genes in the cell. But when we use gutted viruses, most of us use attenuated viruses for safety purposes, and that's a very good thing. We tend to basically remove those genes that deplete the cellular antiviral defenses. And so the specific genetic, cross links there between the cell line and the virus is really what's going to lead to the antiviral signature, and overlaid on top of that, obviously the kinetics and spatial arrangement of how the virus infects. Where is it coming from? Is it transfection based? Is it not? And so I think that those are all things that are easy to ignore, but in reality we shouldn't, because as the poll highlighted, if you're concerned about yield and cost, you need to be thinking about your antiviral defense systems.
Yeah, and I think it's such a good point that you made there, JS, about the cell line virus pairing, right? Because each of these, and even you alluded to it earlier, even within the HEK lineages, each of these is a unique molecular environment. So the chances of having, I'm just gonna put it in like a silver bullet type of approach to address all of this is difficult. And so I'm a big fan of having lots of tools kind of in the toolbox type idea. And then maybe we could talk a little bit about flexibility in manufacturing a little bit later, but having a lot of tools in the toolbox to address these. And I know, there's a lot of talk about genetic modification of cells, siRNA based approaches, small molecules such as the ones we're using, or folks who, in the lenti space, certain processes have HDAC inhibitors, for example. So kind of embracing that small molecule approach. Having all of these different tools available to us, I think is going to allow us to stack strategies, right? Because a lot of these strategies are actually quite complementary. And although, and maybe JS, you can comment on this a little bit, you can't knock out everything, right? There are going to be certain key pieces within the cell you won't be able to knock out. And this is where I think, the small molecule approach or the siRNA approach, although that's transfection and another transfection thing you got to put in there, can really have an advantage over some of those. So, JS, I'm just curious if you want to comment on some of the knockout strategies.
Yeah. I mean, that's kind of the thing, right? I think when you look at the antiviral defense in three parts, you've got the sensors, you've got the signal transducers, and then you've got the effectors. There's a lot of effectors to knock out. So that's not knocking them out one by one is maybe not the most efficient way to go about it. Not to mention that obviously those effectors can have functions in the cells as well that we're not really thinking about. Probably the most efficient way to target is actually in the middle there, the signal transducers. However, those signal transducers regulate so many things in the cells that knocking them out basically leads to cells that don't grow well, cells that die quickly, cells that basically are turned into tumorigenic cells, which the regulators really don't want. And so that's kind of the Pandora's box that you kind of open with targeting those transducers genetically. But that's where kind of the small molecules have an advantage, because that's where we really can have very transient effects. And at the very top, the sensors, well, obviously it's a bit of an hour-shaped glass, right? There's a lot of sensors that you could target, and obviously for some viruses you may be able to restrict it a little bit, but at the end of the day, there's so many phases of the virus replication cycle, and many places where the virus can be sensed, and so it's less efficient of a strategy, I guess, in a way.
Yeah, and I really like that idea of hitting the things temporally and spatially, as you alluded to, like hitting those pathways at the time you need to hit them, so you're not really affecting those ones that are intrinsically linked to cell survival and replication. You can grow your cells to the density you need them to be at, then hit them at the time of infection or just after. It's kind of funny. We talk a lot about moving AAV towards where the antibody folks, hey, we're where the antibody folks were a decade ago, and we'll get there. And I truly believe that we will. But I wonder, and this is just kind of the fun part of the conversation of whether an industry as a whole will start trying to kind of triangulating on a few end-to-end processes, really defined processes that can be deployed by the industry so that there's a little more consistency to the yield and the quality that we get. You know, we talk about all the different lineages. I wonder if we're going to start triangulating on one lineage that's going to be, you know, particular for a certain serotype. But just any thoughts on that?
Yeah, you know, I think that's where we all want to go, right? At the end of the day, I think what we're seeing out there is there's so many different processes. Everybody's got a different subclone. They're all behaving very differently. And so that's really creating a lot of variety out there and preventing us from very closely understanding each process. And so I do think that that's going to get there, but we still have a lot of work to do, particularly in the virus manufacturing space. There's so many different viruses that are being made. It's still hard to triangulate on the cell line that's going to be most effective for all AAV serotypes, all lenti's, etc. So there's still a lot of work to do, no doubt. But you know, again, just talking to the point of clonality, I mean, I think this one is super interesting. You know, I come from the cancer therapeutics background, and when we were thinking about viral sensitizers at the onset, it was all about overcoming tumor heterogeneity to viral infection. And that heterogeneity is playing out in manufacturing subclones as well. And it's more than just, oh, I have this HEC293 variant, you have this other HEC293 variant. You know, clones can drift as you scale, right? When we talk about scalability and you know, high density processes, et cetera, you know, you're starting from a single vial of cells and you're growing that cell, that vial for, you know, for sometimes 2,000 liter batch bioreactors. You know, you've amplified that tremendously. There's significant potential for those clones to drift over time, and their response at the end of the day may be different than the tube you started with, just from drift. And while we're kind of on the point of scale, you know, when we're working in a little shaker flask, you know, if you have a low level contaminant coming from your plasmid, for example, like a little bit of LPS or some endotoxin of some sort, you know, it might be not concentrated enough that you know, it won't trigger any defenses at small scale. But now when you have a 2,000 liter bioreactor, just that low level contaminant is there at high enough of a frequency that it will trigger a response. And those cells communicate, right? Those antiviral defenses start in one place and then they spread. And so all of a sudden you can have a batch that it wasn't as productive for that very low contaminant. And so these are all factors that, you know, we tend to not think about, but that can actually eventually play a role in your process if you're not thinking about it and staying on top of it.
Very
Yeah,
interesting.
yeah. Oh, sorry, Ben. I was just going to comment, like, so many good points there. Like, especially that concept of, you know, the LPS, the PG, even the CPG motifs found within some of these plasmids that we're using. These are also PAMPs, right? And so, and to JS's point, a little bit of PAMP goes a long way, because as much as I talked about that cell being intrinsic and having its own intrinsic ability, to JS's point there, it does start spitting out cytokines and chemokines and communicating with the cells around it. Again, that's part of the evolution here. You know, our body's supposed to be warning the neighboring cells about, you know, mounting an antiviral defense. So as we pack those cells close together and they start communicating, yeah, the dynamics can really change of those cells. Sorry, Ben, it sounded like you wanted to say something.
Yeah, we've been able to talk so far about the context of viral manufacturing when it comes to these antiviral defenses. What about the context of cell therapies? Let's try to keep this a little bit on the brief side, but how could antiviral defenses be impacting cell therapy production?
Yeah, so this is another good point. So obviously, for cell therapy production, you know, there's obviously the manufacturing of the lenti's. You know, it can be helpful there. But the cell therapy itself, when we think about the kinds of cells that we're looking to transduce, and a lot of times again, it's the introduction of nucleic acid, regardless of whether you're using a nanoparticle-based approach or a viral-based approach. And these cells are coming out of you know, sometimes healthy individuals, sometimes sick individuals, but they're still primary cells. And they're cells from the hemopoietic / immune compartment for most part. These are cells that really do not want to be infected, right? And so they most definitely have a fully intact suite of antiviral defenses. So those same pathways we're talking about in this manufacturing context of viral vectors, they're going to be at play trying to prevent those transduction events and the post-transduction events, whether it's integration or maybe a CRISPR-based thing. They are going to be mounting a response to that incoming nucleic acid or that incoming virus. Obviously, a little more complicated there. Maybe JS, you could talk about this a little bit too, is you're much closer to the product though. If you're applying, say, small molecules, this is a place you probably knockout's not going to play too much of a role, I'm not sure. But you're now touching the actual product. So a few more regulatory things to consider there, but certainly would be beneficial to helping increase those transduction efficiencies.
Yeah, I think that's right, JD. You know, transduction of these cell therapies is clearly increasingly an issue, and it's not, you know, something that particularly impacts CAR T manufacturing. I think that transduction efficiency is relatively good, although, you know, you can always do better. But I think as people are starting to get creative with NK CAR and, you know, hematopoietic stem cell based therapies, where those are very, very difficult to transduce, you know, these types of approaches can really help. And again, I'm not sure about the feasibility of the genetic manipulation there. That to me, that is a little bit more challenging in, you know, from a regulatory perspective. But that said, it's not to say that can't work.
Very good. Excellent. So this concludes the section on the fireside chat. So we're going to move to the Q&As in just a minute. But before we do that, we have one final poll. So appreciate you participating in that as well. So we'll bring that up. Do you have an active program or research concerning antiviral defenses in production systems for viral medicines? The options being yes, no, or planned. And again, would really appreciate it if everyone could participate in this, and then we'll look at the results together. So we'll give it another couple seconds here. All right, so most people do not. So hopefully the conversation today will stimulate some internal discussions within your companies to look into this problem. We're seeing some pretty good results when clients are able to actually reduce these antiviral defenses inside of their systems. So hopefully the discussion we've had today can be beneficial with regards to that. All right, so we're going to move on to the Q&As. So again, if you happen to have any questions, we'll try to get through as many of them as we can. So the first question that we're going to go to, oh, this is a really good question. So we've been able to look at antiviral defenses in the context of viral vector production and cell therapies, but then additionally, if you've got an inducible system, for example, where you're making all of the AAVs endogenous to the cell line, does that reduce antiviral defenses?
Yeah, so this is an excellent question. So, you know, certainly lentivirus is, you know, we see some platforms moving towards packaging cell lines. AAV, there are certainly companies pursuing that as well. And so I'll talk about it in two contexts. Sometimes these platforms, you still have to introduce your genome by a transfection-based approach. And so obviously you have incoming nucleic acid there. But even in the fully inducible systems where everything's in that cell, let's think about that second wave of PAMPs we talked about. You're still going to get those byproducts of assembly and replication. And that's still going to set off bells and whistles. Does that molecular environment look different than a transfection-based approach? More likely, yes. Yeah, for sure. But you're still going to be mounting an antiviral response. And this is where I think, you know, a lot of this discussion, understanding the biology and understanding the critical quality attributes, those two things coming together, I think is really going to be informative in helping us develop these types of strategies.
Yeah, I can maybe add a little to that because, you know, it also depends on the system that you're using too. These inducible systems, we'd like to think that they're perfect, but often they're not. And so you might have some leaky promoters. You may have continuous stimulation that you're not aware of right at the onset. So the devil's in the details on the both, you know, the tightness of the system and the kinetics of the upregulation of the different components at which time. And they all, at the end of the day, integrate to a final antiviral signature that that is going to more likely than not impact yield and quality.
Very good. Thank you, gentlemen. So the next question we have here, maybe we'll just get JD to answer this one. What are the key effector antiviral molecules that affect virions?
Oh, well,
Just, answer that in 30 seconds or less, please.
yeah. I mean, as Jay has said, there's hundreds, if not thousands, of interferon stimulated genes that are really setting up that antiviral state. And I think really the key take home message is yes, that's blunting, your overall yield. But I think more importantly, it's blunting the quality / potency of those vectors, because every modification that you make that the cell makes to that capsid or to that genome is going to affect the biology, the underlying biology of that virus that's escaping out of the cell. So, to narrow it down to the most important ones, that's really, really difficult. Just be aware that it's going to go beyond your overall yield and really start drilling in on the quality. JS, I don't know if you have any additional insight.
Yeah, I mean, I think we've already mentioned a few. There's a bunch of cytosine deaminases, adenosine deaminases like the ApoBex, the ADARs, RNase Ls, even SAM-HD, like there's DNTPases out there, TREX, ZAP. There's a whole whackload of them, and there are many homologs and isoforms that we're not even thinking about, right? So there's just a lot, and in fact, the research on antiviral defenses has really increased over the past decade. And I think again, the first thing that it's underscoring is how complex it is and how many interactions between antiviral defenses, stress response, cell survival responses there are, and so tweaking those responses is challenging.
Yeah,
very good.
for sure. And I think that's why sometimes the genetic approaches, like you said, there's so much redundancy, there's so many effectors. That's probably not the area to go after, but I did like the idea of looking more upstream and using the approaches that influence those upstream molecules.
Perfect. All right. So the next question that we have here, should we be looking at other packaging cell lines besides the typical HEC293 to alleviate this problem? Maybe JS, you could take a quick stab at that.
Hey, that's very interesting. I bet you that there are better cell lines out there that I'm fairly confident about. The reality is we're dealing with a lot of of history, right? People started in these cell lines many years ago, and once they've got through regulatory, people don't want to take the risk and they just keep following the same path. And I completely understand why people do this, but it's probably not the best idea, because at the end of the day, we may be stuck on a cell line that may not be the best cell line at the end of the day. For example, you may have an inherently more productive cell line, but it just already has antiviral defenses that are at a higher level. And in reality, if you knock those down completely, you would in the end have a more productive cell line than what the current standard is, right? So we need to start thinking a little bit more flexibly around that. But case in point, the first starting point to that conversation is really understanding the key quality attributes of your product, because if you do have that understanding, you might be able to change your manufacturing process without really changing your product. And so actually at the ISCT conference we attended a couple of weeks ago, I thought this was kind of a concept that resonated with me. We often hear the process is the product. At ISCT, everybody was saying the product is the product. If you understand what your product is, you can change your manufacturing process and still get gains in productivity. And I think that's important to think about. And yeah, I think that's an exciting area for sure. Cell line development is very interesting. But again, the more variance there is out there, the harder time we're going to have to standardize. But at the end of the day, a winner will certainly emerge.
Excellent. Perfect. Well, we've reached our time, gentlemen. Thank you for the very productive discussion. So we're going to wrap up now. But if you'd like to learn more, by all means, reach out to us on our website. We're available on LinkedIn. You can reach out to us by email as well. The details are right there on the slide. I would happy to have additional conversations with you to discuss antiviral defenses in your production systems. All right, with that, we'll turn it over to Aisha, but thank you everyone for coming.
Thank you very much to our panelists for that very insightful presentation. We have now reached the end of the webinar. If we couldn't attend to your questions, the team at Verrica Biotech may follow up with you. Or as Ben mentioned, if you have any further questions, you can direct them to the email address and the other contact information that is displayed on your screen. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is greatly appreciated as it will help us to improve on our webinars. Now, I'm about to send you a link in your chat box, and let me just get that there. There we are. And so you can view the recording of this event on that page, and you can also share this link with your colleagues so that when they register, they can also view the recording there as well. So I do encourage you to do so. Now, please join us in thanking our speakers once again, Dr. Jean-Simon Diallo, Dr. John David DeYoung, and Benjamin McLeod. We hope you found this webinar informative. Until next time, take care and have a great day, everyone.